Hello everyone. Today we will be discussing about 60 questions based on your first and second PUC syllabus, about 15 questions from the first year and about 45 questions from second year. So we move on to the first question. Identify the incorrect statement with reference to taxonomic categories. Mangifera, Solanum, Panthera represent generic epithets. Sapiens, Aestivum, Domestica represent the specific epithets. The first one is generic epithets. Poaceae, Mercedaceae, Anacardiaceae represent family. Primata, Poales, Sapindales represent a class. So which is the incorrect one? So here you have the ranks as well as the endings. So usually when it is a phylum or a division, when it comes to plants, we have phyta. When it comes to class, it is Sida, whereas orders are ales, family is Acae, and so on. So we have the different ones. So if you come here, Mangifera, Solanum, Panthera represent the generic epithets. It is true, isn't it? We have sapiens, Aestivum, Domestica represent the specific. This is generic. This is specific. So this is also the right one. Whereas when you come to Poaceae, Anacardiaceae, this represents your family. This is also true. Primata, Poel, Sapindales represent class. So this is wrong because Primata, Poel, Sapindales are representing your order. So the incorrect one over here is Primata, Poels, Sapindales represent a class. This is the D option is the right one. Moving on to the next question. Chrysophytes are planktons, nectons, benthic organisms, reeds. So we know that chrysophytes are your diatoms. These are golden brown alga, isn't it? So they also include your desmids. Now, what do you mean by planktons? Planktons are organisms which are free floating. So we have the phytoplanktons as well as the zooplanktons. When we have the free floating organisms, when the plants, we call it as the phytoplanktons, and when it is the animals, we call it as the zooplankton. What do you mean by nectons? Nectons are aquatic organisms which swim about, and these are swimming independent of the water current. When you come to the benthic organisms, what do you mean by benthic organisms? Benthic organisms are bottom dwellers. So they will be found at the bottom of the water body. And then we have the reeds. In Kannada, reeds are known as jondugalu. Okay, jondu. So what do you mean by reeds? These belong to the family Poaceae, grass family. Okay, and these are having very thin, long leaves. So what do you mean by a chrysophyte? What are chrysophytes? So chrysophytes are basically planktons. So they are free floating organisms, golden brown alga. Okay, moving on to the next question. A plant is haploid and has thallus level of organization. It has rhizoids and needs water to complete its life cycle. Because male gametes are motile, the plant may be a bryophyte, a pteridophyte, gymnosperm, angiosperm. So here, the plant is a haploid. Here itself we'll come to know what the right answer is, because pteridophyte, gymnosperms, and angiosperms, these are basically diploid in nature. The dominant phase over here is diploid, whereas when you come to bryophyte, this is haploid in nature. Here itself you got the answer that the plant that we are talking about is a bryophyte. It is haploid in nature. So the right answer over here is A, bryophyte. Match the scientific names in column 1 with their common names given in column 2 and choose the correct option. Column 1 has scientific names, column 2 has common names. Meandrina, Fasciola, Bungarus, Aptenodytes, Hippocampus. Column 2, penguin, seahorse, liver fluke, brain coral, and crate. So which is the one? So when you have a multiple, you have your match the following type of question, you should first find out which is your the very thing that you know. First you see if you know any of it, and then you start doing the matching. So here Meandrina. Meandrina is your brain coral. So you have the picture of Meandrina over here. This is Meandrina, which is your brain coral. We have Fasciola. Fasciola is liver fluke. Okay. So we have Fasciola, which is liver fluke. So we have the picture of liver fluke over here. Then we have Bungarus. Bungarus is your crate. It's a reptile. It's a poisonous snake. Then we have the Aptenodytes. Aptenodytes is the penguin, the scientific name of penguin. Then we have Hippocampus. Hippocampus is sea horse. So where do you have this? Options. So the right answer over here is B, wherein we have Meandrina, brain coral, Fasciola, liver fluke, we have Bungarus, which is crate, Aptenodytes, which is penguin, and we have Hippocampus, which is seahorse. So the right option over here is B. Select the mismatch with reference to estivation. Velvet, Calotropis, twisted, cotton, imbricate, cassia, vexillary, China rose. Now, what do you mean by estivation? Estivation is the arrangement of the sepals and petals in the bud condition, isn't it? So we have the different types of estivation over here. So we have velvet, wherein there is no overlapping. We have twisted, wherein on one side it is being overlapped, and on the other side it is overlapping another petal or sepal. We have the imbricate, the quinquecial, and the vexillary type of estivation. So among these, which is a mismatch? So velvet is seen in Calotropis. It is true. Twisted is seen in cotton, that is Gossypium. This is also true. We have imbricate in cassia, which is true. Whereas vexillary in China rose. This is wrong. Why is it wrong? Because vexillary type of estivation over here, we see it in your Papilionaceae family, that is your Fabaceae family, your P family. Okay. Whereas China rose is the other name for hibiscus, Dasawala. Okay. And there it belongs to Malvaceae family, just like your cotton plant. Okay. Just like your cotton, your China rose or hibiscus belongs to Malvaceae family. So it is not belonging to Fabaceae family, and the arrangement of your sepals and petals is not vexillary type. So the mismatch over here is D, which is vexillary China rose. With reference to the reproductive system of cockroach, match column 1 with column 2 and choose the correct option. Column 1, telomere, gonophore, spermatophore, ovarioles. Column 2, chain of developing ova, bundles of sperm, opening of the ejaculatory duct, the external genitalia. Now this seems to be a little easier. Okay, so here, this itself, if you see ovarioles, chain of developing ova, because here itself, ova and ova. Here you are able to get the answer well in advance. But the problem, if you see with the options that you have over here in the matching, is all have D has P. Okay, so now it's a little confusing. So go for the next one that you know. So most probably the one that will give out the answer to you is your spermatophore, which is your bundles of sperm. Okay, spermatophores, which is bundles of sperm. So here itself you may get the answer. And here, what do you mean by phallomere? Phallomere is the external genitalia when it comes to the cockroach, whereas gonophore is the opening of the ejaculatory duct. So where do you find this? Option among the four, A, B, C, D, where do you find this? So the right answer you can see in B. Okay, so in B, you get the right answer. So the right answer or the right option over here is B. Okay. Select the correct option with respect to the arrangement of cells in an ascending order of their size. So they have asked for the ascending order. What do you mean by ascending order? From the lower to the higher. So you're climbing up the stairs over here. Okay, you are climbing up the stairs. You're not coming down. If you're coming down, it is descending order. So ascending. So you have to start from the smallest to the biggest that is there over here. Now, which are the options do you have? We have the bacteria, human RBC, mycoplasma, and ostrich. So in which order do you get? Now, what is the size of a bacterium? The size of a bacterium may be somewhere from 0.3 to around 8 microns. Okay, whereas when you come to human RBCs, the size of the human RBC may be somewhere from around 7.5 to 8.7 microns. The mycoplasma, one of the smallest organisms that we know, it will be having a size of around 0.2 microns. Whereas the ostrich egg, the largest known single cell, is your ostrich egg, which may be around 15 centimeters approximately. So if you're going for the order, in the ascending order, which is the one? So definitely the smallest one is your mycoplasma, followed by the bacteria, human RBC, and the ostrich egg, isn't it? So here we have the sizes, the difference. So largest one that we have is your ostrich egg. So based on the sizes, if you see here, the option over here is B, mycoplasma, bacteria, human RBCs, and the ostrich egg. Moving on to the next question. Which of the following is not an aromatic amino acid? Tyrosine, phenylalanine, tryptophan, and lysine. Now, first thing, what do you mean by an aromatic amino acid? Aromatic amino acid is an amino acid with an aromatic ring. So we have about 20 amino acids. We have the essential and non-essential. And each one is divided into different types, isn't it? So here we have the aromatic amino acids. So there are 3 amino acids which are aromatic. That is, we have the phenylalanine, tyrosine, and tryptophan. So definitely these 3 are aromatic amino acids, whereas lysine is a non-aromatic amino acid. Actually, it is a hydrophilic basic amino acid, whereas your aromatic amino acids are hydrophobic aromatic, whereas this is hydrophilic. Lysine is hydrophilic basic amino acid. So the right answer over here is lysine. So D, lysine is the right option, which is not an aromatic acid. Select the correct statement about G1 phase. Cell is metabolically inactive. DNA in the cell does not replicate. It is not a phase of synthesis of macromolecules. Cell stops growing. So you know about the cell cycle. So the cell cycle is divisible into the G1 phase, the S phase, G2 phase, the M phase, and we have a very short quiescent phase known as your G0 phase. So what happens? And each phase has its own function. What is happening during those things? So usually during the G1 phase, your preparation of your DNA synthesis is taking place. So nucleotides and all are macromolecules and all are produced. Whereas during the S phase, DNA replication takes place. During the G2 phase, it is preparing for the next phase, that is the M phase, wherein a preparation for mitosis is taking place. Whereas during the M phase, your mitosis or your meiosis, that is the actual division, is taking place. So here, which is the correct statement about G1 phase? We want to find out the right statement. So the first sentence is cell is metabolically inactive. No, it is active. G1 is an active stage, so it is not metabolically inactive. DNA in the cell does not replicate. True. This is true. DNA does not replicate because DNA replication takes place during the S phase and not during the G1 phase. So this is the right answer. Now the next one, it is not a phase of synthesis of macromolecules. No, it synthesizes macromolecules during this step, wherein it is preparing for your DNA replication that is taking place during the S phase. So here your nucleotides, the protein and all are synthesized. Now the cell stops growing. No, it will not stop growing. It is growing, especially the nucleus. During the G1 phase, the nucleus is growing. So among the four, the correct statement that we have is option B. That is, DNA in the cell does not replicate. Who provided the evidence that glucose is formed during photosynthesis and is then stored in the form of starch? Julius von Sass, Engelmann, Van Helmont, and Melvin Calvin. So here we have some scientists who have contributed towards the photosynthesis. So we have Joseph Priestley, who actually focused on the solar rays. Okay, and he was the one who called the gas that is produced as the oxygen. And then we have Engelmann, Theodor Wilhelm Engelmann, who he discovered the effect of varying wavelengths of light on the photosynthesis. And then we have Melvin Calvin, who traced the path of the carbons in photosynthesis, and he was the one who founded the Calvin cycle. Then we have Emerson and Arnold, who stated that the photosynthesis occurs by hundreds of chloroplasts cooperating with each other. Then we have Van Helmont, who conducted experiments which led him to conclude that trees consumed water and not soil. So among these, who was the one who gave us the evidence that glucose is stored in is formed during photosynthesis, stored in the form of starch? It was Julius von Sass. So Julius von Sass, we are also familiar with him because of his works on hydroponic technique. So he was the one who actually gave us the evidence that the glucose that is formed during photosynthesis is stored in the form of starch. The final hydrogen acceptor in electron transport system is oxygen, hydrogen, ubiquinone, or cytochrome C. So here we have the electron transport system, and in the electron transport system we can see that the final acceptor of your hydrogen over here is your oxygen, who combines to form your water. So the right answer over here is oxygen. So A, oxygen is the right answer. What is the amount of oxygen released to the tissues by blood on passing from lungs? 15 ml of oxygen per 100 ml of blood, 70 ml of oxygen per 100 ml of blood, 5 ml of oxygen per 100 ml of blood, 20 ml of oxygen per 100 ml of blood. So here, how do you calculate what is the amount of oxygen that is released to the tissues by the blood? So the blood will be having your oxygenated as well as your deoxygenated blood. So we have the normal arterial oxygen content, okay, which is about 20 ml per 100 ml of blood, and a normal venous oxygen content, which is usually about 15 ml of per 100 ml of blood. So how do you calculate how much is given? Okay, you have the arterial blood as well as the venous blood, isn't it? So how much are you going to, each tissue is going to get? since the arterial oxygen is about 20 ml and the venous is about 15 ml, you have to minus the arterial blood oxygen and with your venous blood oxygen. So definitely it is somewhere around 20 minus 15, that is equal to 5 ml. So each tissue is getting approximately 5 ml of oxygen per 100 ml of blood. Okay. So the right answer is C, 5 ml of oxygen per 100 ml of blood. Which of the following is showing the correct sequential order of vertebrae in the vertebral column of human beings? Cervical, lumbar, thoracic, sacral, coccygeal. Cervical, thoracic, sacral, lumbar, coccygeal. Cervical, sacral, thoracic, lumbar, coccygeal. Cervical, thoracic, lumbar, sacral, coccygeal. So here you can see, we all know that the first vertebrae that we have is your cervical, whereas the last one is your coccygeal. So a little confusion over here, which comes where, isn't it? In the vertebral column, this is where you're getting a little confused. So we'll just see where it is. So if you see the vertebral column, we see that initially we have about 7 bones, which your vertebral column has about 7 vertebrae which belong to your cervical. We have about 12 which forms your thoracic, 5 which forms about your lumbar. Then we have the sacrum, wherein we have about 5 which is fused, and then we have the coccyx which is once again fused. So the right order you can see, it begins from the cervical, which is about 7 vertebrae, followed by 12 thoracic vertebrae, 5 lumbar, 5 fused sacrum, and then we have the coccyx which is also fused. So the sequence is cervical, thoracic, lumbar, sacrum, and the coccyx. So which is the right option over here? We have D, which is in the correct sequence over here, wherein we have cervical, thoracic, lumbar, sacral, and coccygeal. Excessively high heart rate can reduce cardiac output because blood is moving too fast through the lungs to pick up enough oxygen. It tires out the heart muscles, so they pump slower. It reduces the time for ventricular filling, which reduces the stroke volume. PR interval increases, which leads to longer ventricular diastole and shorter ventricular systole. So here, what do you mean by the PR interval? PR interval is nothing but your atrial, the time that you have between the atrial and the ventricular depolarization. So among these 4, which is the right option? So the right option over here is your C. It reduces the time for ventricular filling, which reduces the stroke volume. So whenever there is excessively high heart rate, the cardiac output reduces mainly because the stroke volume decreases. Okay, because there is a reduced time for the ventricles to be filled. So what do you mean by stroke volume? Stroke volume is the amount of blood pumped from the ventricles per heartbeat. So definitely when the stroke volume is less, definitely the cardiac output is also reduced. Yeah, so the right answer, the right option over here is C. That is, it reduces the time for ventricular filling and which reduces your stroke volume. Select the option that correctly matches the structures with its location and function. Eustachian tube, anterior part of the internal ear. A function is equalizes air pressure on either sides of tympanic membrane. Cerebellum, location is midbrain, brain controls respiration and gastric secretions. Hypothalamus, location is forebrain, function is it controls body temperature, urge for eating and drinking. Blind spot, location is near the place where the optic nerve leaves the eye, and the function is rods and cones are present but inactive over here. So we'll see here, eustachian tube, anterior part of the internal ear. No, it is a part of the middle ear. Okay, function equalizes the air pressure on either sides of the tympanic membrane. Then we have cerebellum, which is the part of the midbrain. No, it is a part of the hindbrain. Okay, it is a part of the hindbrain. That is rhombencephalon. Okay, so this is wrong. It controls respiration and gastric secretion. No, the respiration and gastric secretion is controlled by your medulla. Okay, it is controlled by your medulla, whereas your cerebellum controls your posture, your equilibrium, as well as your muscle tone. When you come to hypothalamus location is forebrain. It controls the body temperature, urge for eating and drinking. So this is the right option. And the blind spot. So here it is given as near the place where the optic nerve leaves the eye. Actually, the optic nerve lives from this place, from the blind spot. It starts from that place actually. And whereas rods and cones are present but are inactive here. No, here the rods and cones are completely absent. So the correct match over here is C, wherein we have the hypothalamus located in the forebrain, which controls the body temperature, urge for eating and drinking. Choose the option which shows two plants in which new plantlets arise from the same organ. Dahlia or dahlia and ginger, potato and sweet potato, dahlia and rose, potato and sugarcane. Now the question is, you have to find two plants in which plantlets arise from the same organ of the plant. Now if you take dahlia or your dahlia, we have the tubers over there, which is obtained from your, which is nothing but your roots. Okay, so this is roots. Whereas ginger, shunti, ಅಲ್ಲಿ ಹೊಸ ಗಿಡ ಎಲ್ಲಿಂದ ಬರ್ತದೆ? It is from your stem, isn't it? So ginger is a rhizome. So it is a modified stem actually. So ಈ ginger ಇಂದ, ಆ ಶುಂಠಿಯಿಂದ, ಆ stem ಇಂದನೆ ನಮಗೆ ಹೊಸ ಗಿಡಗಳು ಬರ್ತದೆ. So you can see here, okay, ಆ stem ಇನ nodes ಇಂದ ಹೊಸ ಗಿಡಗಳು ಬರ್ತದೆ. It's a rhizome. So stem, ಇದು stem. So E answer alla, definitely, isn't it? Yake? They have asked for same organ inda irbeku. So root matte stem eradu same organs alla. So this is not the option. The next option you have is a potato and sweet potato. Potato, nimma batategalu. Okay, sweet potato, genasu. So here you have. Now, if you take a potato is a stem. Once again, it's a stem tuber. It's a modification. It's a modified stem. So allinda adu barta ide. Hosa gidagalu arise ಆಗ್ತದೆ. ನಿಮಗೆ ಇಲ್ಲಿ ಕಾಣ್ತಾ ಇದೆ ಹೇಗೆ ಅದು ಚಿಗುರಿದೆ ಅಂತ. Whereas when you come to your sweet potato actually grows from your adventitious roots. So it's a adventitious root modification. So definitely idu kuda option alla. ಯಾಕೆಂದ್ರೆ ಒಂದು stem modification, ಇನ್ನೊಂದು ನಿಮ್ಮದು root modification. Coming to the third option, dahlia and rose. ಮೊದಲು ಹೇಳಿದಾಗೆ, as we have already discussed, dahlia or dahlia is a flower, isn't it? So this dahlia or dahlia that is there, it grows from your tubes. So here we have, they are the root modification. They come from the roots. Whereas the rose, usually gulabi ಗಿಡಗಳು, from where do you get? The rose ಗಳಲ್ಲಿ usually ನಾವು ಆ ಸ್ಟೆಮ್ ಕಟ್ಟಿಂಗ್ಸ್ ಅನ್ನು ತಗೊಳ್ತೇವೆ, ಎಸ್ಪೆಷಲಿ ಆ ನೋಡಲ್ ರೀಜನ್ ಇಂದ ತಗೊಳ್ತೇವೆ. ಸೊ ಆ ನೋಡಲ್ ರೀಜನ್ ಅಲ್ಲಿ ನಮ್ಮ ಇಂಟರ್ಕ್ಯಾಲರಿ ಮೆರಿಸ್ಟಮ್ ಇರುವುದರಿಂದ ಅಲ್ಲಿಂದ ಹೊಸ ಗಿಡಗಳು ಬರ್ತದೆ. ಸೊ ಅಲ್ಲಿಂದ ಹೊಸ ಚಿಗುರು ಬರ್ತದೆ. ಸೊ ನಿಮ್ಗೆ ಯಾವ್ದಾದ್ರು ಗಿಡ ಅಲ್ಲಿ, ನಿಮ್ಗೆ ಗುಲಾಬಿ ಗಿಡದಲ್ಲಿ ಫ್ರೂಟ್ ಸಿಗುದಿಲ್ಲ, ಬಿಕಾಸ್ ಇದು ನಮ್ಮದು ಕಲ್ಟಿವೇಟೆಡ್ ವೆರೈಟಿ. ಸೊ ನಿಮ್ಗೆ ಅಲ್ಲಿ ಅದು ಆರ್ನಮೆಂಟಲ್ ವೆರೈಟಿ, ಕಲ್ಟಿವೇಟೆಡ್ ವೆರೈಟಿಯಲ್ಲಿ ನಿಮ್ಗೆ ಫ್ರೂಟ್ ಸಿಗುದಿಲ್ಲ, ಆಸ್ ವೆಲ್ as ಅದರಲ್ಲಿ ಸೀಡ್ ಸಿಗುದಿಲ್ಲ. ಸೊ ನಿಮ್ಗೆ ಒಂದು ಹೊಸ ಗಿಡ ಬೇಕಾದ್ರೆ ನೀವೇನ್ ಮಾಡ್ತೀರಿ, ಆ ಸ್ಟೆಮ್ ಅನ್ನು ಕಟ್ ಮಾಡಿ, ಆ ಸ್ಟೆಮ್ ಯಾವ ರೀಜನ್ ಅಲ್ಲಿ ಕಟ್ ಮಾಡ್ತೀರಿ, ಇಂಟರ್ನೋಡಲ್ ರೀಜನ್ ಅಲ್ಲಿ ಕಟ್ ಮಾಡೋದಿಲ್ಲ. ನಾವು ಕಟ್ ಮಾಡೋದು ಅದಕ್ಕೆ ಆ ನೋಡ್ ಇರುವ ಹಾಗೆ ನೋಡಿ ನಾವು ಅದನ್ನು ಹೋಗಿ ನೆಡ್ತೇವೆ. ಹಾಗೆ ಏನಾಗ್ತದೆ, ಅಲ್ಲಿಂದ ಅದು ಚಿಗುರು ಬರ್ತದೆ. isn't it? ಅಲ್ಲಿಂದ ನಮ್ಗೆ ಅದರದ್ದು ಚಿಗುರು ಬರ್ತದೆ. ಸೊ ಡೆಫಿನೆಟ್ಲಿ ಇದು ರೂಟ್, ಇದು ಸ್ಟೆಮ್. ಸೊ ಇದು ಕೂಡ ಆಪ್ಷನ್ ಅಲ್ಲ. ವಿಲ್ ಕಮ್ ಟು ದ ಲಾಸ್ಟ್ ಒನ್, ಪೊಟೇಟೊ ಅಂಡ್ ಶುಗರ್ ಕೇನ್. ಸೊ ಪೊಟೇಟೊ ಆಸ್ ವೆಲ್ as ಶುಗರ್ ಕೇನ್ ಅಲ್ಲಿ ಆ ನೋಡಲ್ ರೀಜನ್ ಅಲ್ಲಿ ನಮ್ಗೆ ಹೊಸ ಗಿಡಗಳು ಬರ್ತದೆ. ಸೊ ಪೊಟೇಟೊ, ಬಟಾಟೆ, ಶುಗರ್ ಕೇನ್, ಓಕೆ, ಇಲ್ಲಿ ಕಬ್ಬು, ಅದರಲ್ಲಿ ಕೂಡ ನೀವು ನೋಡಿ ಆ ಗಂಟು ರೀಜನ್ ಅಲ್ಲಿ ವಿಚ್ ಇಸ್ ನಥಿಂಗ್ ಬಟ್ ಯೋರ್ ನೋಡಲ್ ರೀಜನ್, ಆ ಗಂಟು ರೀಜನ್ ಅಲ್ಲಿ ನಮ್ಗೆ ಹೊಸ ಚಿಗುರುಗಳು ಬರ್ತದೆ. ಸೊ ವಿಚ್ ಇಸ್ ದ ರೈಟ್ ಆನ್ಸರ್ ಓವರ್ ಹಿಯರ್. We are supposed to find the two plantlets which arise from the same organ. So the right option over here is potato and sugarcane. Potato and sugarcane is the right option. Next question. Select the correct statement with respect to the reproductive cycles in mammals. A. Estrous cycle occurs in primates. B. Menstrual cycle occurs during favorable seasons. C. Females in estrous phase are said to be in heat. D. Menstrual cycle occurs in non-primates. So we have to find the correct statement over here, isn't it? And the correct statement, as it is already given, is females in estrous phase are said to be in heat. Now, why is this statement right? Why not the first one? Okay, so here we have, sorry, excuse me, I'm sorry. Okay, so here you can see estrous phase, estrous cycle occurs in primates. So we know that this occurs in non-primates and not in primates. Menstrual cycle occurs during favorable seasons. No, menstrual cycle is seen in primates who are continuous breeders, whereas here the thing is favorable season. It is not seasonal breeders. The mammals which are showing here menstrual cycle are continuous breeders and not your seasonal breeders. So definitely that is not the right answer. C is the right answer, wherein females in estrous phase are said to be in heat. D is not the right answer because menstrual cycle occurs in primates and not in non-primates. Read the following statements and choose the correct option. Statement 1. Chances of survival of young ones is greater in viviparous organisms. Statement 2. All mammals are viviparous. The options are both statement 1 and 2 are correct. Statement 1 is correct, but statement 2 is incorrect. Statement 1 is incorrect, but statement 2 is correct. And D, both statements 1 and 2 are incorrect. Now we will see the first statement. Chances of survival of young ones is greater in viviparous organisms. Okay, it is greater in viviparous organisms, and we know that this is right, isn't it? We know that this is right. Whereas statement 2, all mammals are viviparous. This is no, this though mammals are viviparous, there are some exceptions to it. For example, we have the duck-billed platypus as well as the echidna. So we have the duck-billed platypus as well as the echidna, spiny echidna, which are not viviparous. They don't give birth to the young ones directly. Instead, they are oviparous. So definitely, all mammals are not viviparous because we have exceptions over here. So statement 1 is right and statement 2 is incorrect. So here, the right answer that you have is statement 1 is correct, but statement 2 is incorrect. Moving on to the next question. Anther wall consists of 4 wall layers where tapetum lies just inner to the endothecium. Middle layers lie in between endothecium and tapetum. Endothecium lies inner to the middle layers and tapetum lies next to the epidermis. So if you take a section of your anther wall, we can see here that the outermost layer is the epidermis. This is followed by a layer of endothecium, and then we have the layers of middle layers we have, isn't it? We have the middle layers. So here, next to the middle layers, we have the microspore mother cells. the tapetum, and next to the tapetum is the microspore mother cells. So here definitely the question is the anther wall consists of 4 layers where? Okay, so we have from epidermis, then we have the way it comes inside. So definitely the right answer over here is B, where the middle layer lies between the endothelium and the tapetum. So the right answer over here is B. That is, middle layers lie in between endothelium and tapetum. Next question. Even in the absence of pollinating agents, seed setting is assured in Commelina, Zostera, Salvia, and Helianthus. So here we have, this is the Commelina, we have the Zostera, Salvia, and Helianthus. Helianthus is nothing but your sunflower. So among these, in which even in the absence of your pollinating agent, you are going to have a seed setting. So that means pollination is going to take place. So definitely here if you see, Commelina is one plant. Okay, Commelina, that is it. Now this is a cleistogamous plant. So definitely it open, even before the buds open, they remain closed, and when they are closed itself, within that only self-pollination will be taking place. So they don't open. Even before they are open, pollination has already taken place. Commelina. Whereas when you come to Zostera, water is the pollinating agent here. Salvia and Helianthus, insects are the pollinating agents. So for this, you need the pollinating because they are chasmogamous flowers. They open up. It is only the Commelina among these 4 options that you have, which is a cleistogamous flower. That is, it is a closed flower. So definitely you are going to have seed setting is seen even if the pollinating agents are absent, because in Commelina you get to see self-pollination. Okay, whereas that is autogamy. Whereas in the other 3 you get to see your cross-pollination. So the right answer over here is Commelina. Next question. The total number of nuclei involved in double fertilization in angiosperms are 2, 3, 4, and 5. So how many nuclei are involved? So definitely we know that we have 2 male nuclei. One will come and fuse with the egg cell, and the other one will fuse with the 2 polar nuclei. So you have to count how many nuclei are involved. So if we count, we know that there are 2 male nuclei. So 2 are done, isn't it? And the others that are there, we have 1 egg cell, so 3 nuclei, haploid. Then we have 2 polar cells, polar nuclei. So a total of 5 nuclei are involved. So out of this, the right option that you have is 5. I hope you have understood. How did we get 5 over here? It is not 2, 3, 4. It is 5 nuclei because we have 2 male nuclei. Then we have 1 egg cell and 2 polar nuclei. So a total of 5 nuclei are involved over here. A dicot plant bears flowers but never produces fruits and seeds. The most probable cause may be the plant is dioecious and bears only pistillate flowers, monoecious, dioecious and bears only staminate flowers, and the plant is bisexual. So first thing you should know, what do you mean by a dioecious plant? A dioecious plant is a plant where you have separate male and female plant, as in case of your papaya. Okay, as in case of papaya, where you are, as well as your date palms, where you have separate male and separate female plants. You don't have the male flowers and female flowers in the same plant. Whereas when you come to monoecious plant is a plant where you have the male and female flowers in the same plant, as in case of your coconut palm, as well as your cucurbitaceae family, cucurbita, cucumber family, which are monoecious plant. Male and female flowers are found in the same plant. And when you say dioecious plant, I said you have a separate male plant and a separate female plant. So if the male plant has male flowers, then it is known as a staminate plant. And if it has got female flowers, it is known as a pistillate plant. So we will, pistillate flowers are, they will just see a dicot plant bears flowers but never produces fruits and seeds. The most probable cause may be definitely It has flowers, but it is not producing fruits and seeds. Now, if it is a monoecious plant, wherein both the male and female flowers are there, it can produce, because pollination can take place over there. So it can produce the fruits over there, isn't it? Because female flower is also there. In case of a bisexual flower, bisexual flower is a flower wherein both the androecium and gynoecium is present over there in the same plant, isn't it? In the same flower. So even in a bisexual flower, because the female flower is there, there is a chance of production of fruits as well as seed. Whereas here the option, so definitely it cannot be a monoecious plant, it cannot be a bisexual plant. Now the options that you have is, it is a dioecious plant bears only pistillate flowers, and dioecious plant which bears only staminate flower. Now, when it has pistillate flower, it indicates that it is a female flower. So since the female flower is there, there is a chance of pollination taking place, and their fruits and seeds are produced. Whereas when it comes to a staminate flower, it has in it. It does not have an ovary, so there's no chance of fruit setting. Okay, so here the staminate flowers will just be donating its pollen grains to a female flower or to another flower, isn't it? But it will not produce your fruits and seeds because it does not have an ovary. So definitely the plant that we are talking about should be dioecious, and it bears only staminate flowers. Staminate flowers means male flowers. It has only male flowers. So the option, right option over here is C. A sex gland which contributes fluid containing sugar fructose that provides spermatozoa energies for swimming is corpus gland, prostate gland, seminal vesicle, bulbourethral gland. Now, what is the function of these glands? We have the corpus gland, the prostate gland, the seminal vesicle, as well as the bulbourethral gland. So remember, corpus gland, the other name for corpus gland is bulbourethral gland. It is the same one. It is the other name. Corpus gland and bulbourethral gland is the same gland. Now, a organism, a man, a human being has this, male has these glands, isn't it? So what is the function? Now, if you take the seminal vesicle is actually the gland which secretes fructose and most of the seminal fluid. Whereas when you come to the prostate gland, a single gland is there. It secretes a watery alkaline fluid to raise the pH of the vagina. Whereas the bulbourethral gland, you have a pair which is also known as a corpus gland. It secretes a thick mucus which acts as a lubricating agent. So these three actually contribute towards your secretion of your seminal plasma, seminal fluid, isn't it? So here, which is the one which actually contributes the fluid which is having sugar fructose in it? So among these glands that we have, it is the seminal vesicle. Okay, so the option is C, seminal vesicle, which actually secretes the fructose and most of the seminal fluid. Milk secreted from the cells of alveoli of mammary lobes reaches nipple through lactiferous duct, which is represented as L, mammary duct, which is represented as M, mammary tubule, which is represented as T, and mammary ampulla, which is represented as A, in the following order: TMAL, MTLA, MTAL, ATML. So if you see, from where does the milk flow, in which order does it flow? So definitely from the alveoli of the mammary lobes, it has to reach the nipple, isn't it? From here the milk has to come till here. So which path does it take? So actually the milk from the alveoli moves into the mammary tubule, and from the mammary tubule, so this one, from the mammary tubule, so that is T. From the mammary tubule, it goes into your mammary duct, so which is M. Then we have it moving into your mammary ampulla, and then ultimately it comes to the lactiferous duct L, and from there it will move into the nipple. So the right option over here, the flow of milk from the alveoli of the mammary glands to the nipple, it is through TMAL, where T represents your mammary tubule, M represents your mammary duct, A represents your mammary ampulla and L represents your lactiferous duct. So the right option over here is A, TMAL. The process of release of spermatozoa from Sertoli cells into cavity of the seminiferous tubules is called spermiogenesis, spermatogenesis, spermatocytogenesis, spermiation. So, what is the process of release of spermatozoa? Spermatozoa from the Sertoli cells. So, spermatozoa is the sperms, isn't it? So, what is that process known as? So, we have four options over here. We have spermiogenesis, spermatogenesis, spermatocytogenesis, and spermiation. So, which is the right answer? Now, first, you should know what spermiogenesis is. Spermiogenesis is a process wherein the non-motile spermatids are being transformed into your motile sperms or your spermatozoa, whereas spermatogenesis is a process of production of the sperms from the primordial germ cells, and spermatocytogenesis. So this is actually the production of the stem cells. Now this is nothing but your production of your spermatogonial cells. Okay, and spermiation is actually the release of the sperms from the Sertoli cells into the cavity of the seminiferous tubules. So spermiation is the right option. That is option D is the right option. That is spermiation is the process of release of spermatozoa from Sertoli cells into the cavity of the seminiferous tubules. If mammalian ovum fails to get fertilized, which one of the following is unlikely? Corpus luteum will disintegrate, progesterone secretion rapidly declines, estrogen secretion increases, primary follicle starts developing. So the right answer over here is C. Estrogen secretion increases. Now, why did we choose this as the right answer? Now, what does the question state? The mammalian ovum, that is, it has failed to get fertilized. That means it is not fertilized. So, there's no chance of pregnancy. So, definitely, which is unlikely. Okay, which is not going to, which is very rare to happen, which is not going to happen. So, normally. So, we definitely, corpus luteum will disintegrate. If there is no ovulation, fertilization taking place, then definitely the corpus luteum will gradually disintegrate and it will make way for the next cycle, next menstrual cycle. Then the progesterone secretion rapidly declines. Definitely the secretion declines away of the progesterone because progesterone and estrogen is secreted by the corpus luteum, isn't it? So when the corpus luteum is getting disintegrated, definitely progesterone secretion also declines, whereas primary follicle starts developing. Once the corpus luteum completely disintegrates, then definitely for the next cycle, because primary follicle is actually the precursor of the Graafian follicle, isn't it, which will then form into your corpus luteum after the ovulation process. So definitely this also is going to likely happen. Now, which is not likely to happen, so unlikely it is given. Estrogen secretion increases. No, if the corpus luteum is disintegrating, definitely the corpus luteum is secreting your progesterone and estrogen also. So since it is disintegrating, the estrogen and the progesterone concentration also is going to decline. So the right answer over here is C. Estrogen secretion increases because it is unlikely to happen. The first case of IVF-ET technique success was reported by A. Louis Joy Brown and Patrick Steptoe and Robert Edwards, Robert Steptoe and Patrick Edwards, Louis Joy Brown and Robert Edwards. So, which is the first case of IVF-ET, in vitro fertilization embryo transfer, isn't it? So, the first IVF baby, the commonly known as test tube baby, was born in 1978, and the first baby to be born, the first test tube baby, successfully be born was Louis Joy Brown. So definitely she is the baby, and she was not the one who developed herself into a test tube baby, isn't it? So definitely that is not the right answer. So which is the right option over here? So we have Patrick Steptoe and Robert Edwards. Okay, we have Robert Edwards and Patrick Steptoe, who actually were able to get the first successful IVF baby born. That is your Louis Joy Brown. Okay, in 1978. So the right option over here is B, Patrick Steptoe and Robert Edwards. Which of the following contraceptive method correctly matches with its mode of action? Contraceptive method: tubectomy, oral pills, diaphragms, IUDs. Mode of action: make the uterus unsuitable for implantation, inhibit ovulation and implantation, spermicidal, and increases phagocytosis of sperms within the uterus, blocks gamete transport. So what is asked over here? Which of the contraceptive method correctly matches with its mode of action? Among the four, which is the right option over here? So here, what is tubectomy? Okay, what is the mode of action of tubectomy? It actually blocks the gamete transport. Okay, it will be blocking the gamete transport. So definitely this is not the right answer. Oral pills. Now, what is the function of the oral pills? It inhibits ovulation and implantation. So definitely this is right, isn't it? Now, what is the function of the diaphragms? Here it is given as it is spermicidal and increases phagocytosis within the phagocytosis of sperms within the uterus. This is not the right answer because diaphragms usually prevent the sperm from reaching an ovum, and IUDs that are there, they block the gamete transport. No, they don't block the gamete transport. Actually, what is happening is they increase the phagocytosis of sperms within the uterus. So among this, which is the right option? So definitely it is B, isn't it? We have B, oral pills inhibit ovulation and implantation. On crossing two heterozygous tall plants, TT, a total of 500 plants were obtained in F1 generation. What will be the respective number of tall and dwarf plants obtained in F1 generation? So, what is this? So, here you can see it has been given as heterozygous plant, capital T, small t, isn't it? So, here we have worked out the cross. So, here you can see when these are the parents, and these parents will be giving you two different types of gametes, and it is a chance factor, as you know, and when these gametes will start fusing, we'll get the plants in this order. We are going to get 3 tall plants and 1 dwarf plant. So we are getting it in the ratio of 3:1 ratio. So 3:1 ratio, if you go, we have 500 plants, isn't it? We have 500 plants that are obtained in F1 generation. So out of the 500 plants divided by 4, so you are going to get 3:1 ratio, isn't it? So how much is it? So definitely here it is 375 and 125 plants. So the right option over here is 3A, 375 and 125 plants. So 375 will be tall plants and 125 will be dwarf plants. Due to non-disjunction of chromosomes during spermatogenesis, some sperms carry both sex chromosomes 22A plus XY, and some sperms do not carry any sex chromosome 22AA plus 0. If these sperms fertilize normal eggs 22A plus X, what type of genetic disorders respectively appear among the offsprings? Turner syndrome and Klinefelter syndrome, Klinefelter syndrome and Turner syndrome, Klinefelter syndrome and Edwards syndrome, Turner syndrome and Edwards syndrome. Now, what is this? Now, we know that Turner syndrome is a condition where there is a lack of sex chromosome. One sex chromosome is less. So, it is usually 44AA plus X0, isn't it? We have, when it comes to your sex chromosomes, this is X0. We do not know which chromosome is missing, whether it is an X chromosome or a Y chromosome is missing, because only one X is there. Usually they will be showing the feminine characteristics because there is X in it. Whereas when you come to Klinefelter syndrome, we have an extra sex chromosome. So we have XXY. isn't it? Now, which is extra, we do not know, but we have an extra sex chromosome over here. Whereas when it comes to Edwards syndrome is because of the trisomy of the 18th chromosome. What do you mean by trisomy? We have an extra 18th chromosome over here. One extra copy is, instead of having 2, we have 3 18th chromosome. Whereas, so here among these, now definitely it cannot be this and this, isn't it? Because here we have Edwards syndrome. So this has got nothing to do with the sex chromosome. So not these two. Whereas, which is the one? Whether it is Turner syndrome and Klinefelter syndrome or Klinefelter syndrome and Turner syndrome. Now what is given over here? We have both sex chromosomes, isn't it? 22A plus, we'll just concentrate here. We have 22A plus XY. We'll just concentrate on the sex chromosomes. So one has got XY, the other one has got 0, isn't it? So when this goes around, we should get somewhere where we have the one which does not have a sex chromosome and the other which has a sex chromosome. So this is somewhere where this is going and binding with a egg which has a X chromosome. So what are you going to get over here? So definitely it is going to be Klinefelter syndrome and Turner syndrome, because this egg will, if it fuses with this one, you are going to get 44AA plus XXY, which is your Klinefelter syndrome. And if it is going to fuse with this sperm, then it is going to be 44AA plus X0, isn't it? So definitely it is Klinefelter syndrome and Turner's syndrome. Which of the following will not result in variation among siblings? Independent assortment of genes, crossing over, linkage, and mutation. Now, what is asked? Where there are no variations among siblings. So, definitely we know that though we belong to the same parents, your sibling, for example, if you have a brother or a sister, you all belong to the same parent, you have similarities among you, but still you have variations, isn't it? We can identify. You all are siblings, but there is variation. Why there is variation? Definitely because of crossing over. So crossing over, where you have the meiosis taking place, because of that crossing over, you definitely have a lot of changes among you, though you belong to the same parent. You know, why is it we sometimes, maybe when you were very young, you thought, why is it that this is my brother, you know, same parent we have, but maybe I am adopted or my brother or sister is adopted, but now you know it is not so. It is mainly that variations that you get to see is because of crossing over. So among these 4, which one is not going to lead to variations? Definitely independent assortment of genes will lead to variations. Crossing over that is seen during meiosis is going to lead to variations. Linkage is not going to contribute towards the variations among the siblings, but mutation is also going to contribute towards your variations. So among the 4, the right option is C, linkage. Mother and father of a person with O blood group have A and B blood group respectively. What would be the genotype of both mother and father? Mother is homozygous for A blood group and father is heterozygous for B. Mother is heterozygous for A blood group and father is homozygous for B. Both mother and father are homozygous for A and B blood group respectively. Both mother and father are heterozygous for A and B blood group respectively. Now what is given? Mother and father. So we have the mother and the father, isn't it? Now mother has A blood group and father has B blood group, and they are going to give you a child, an offspring with O blood group. Now if you see the genotype of O, we know that this does not have any antigen. So this genotype you can see as I0, I0, isn't it? We represent it this way. So definitely this should be present in the parents. It should be present in the parent, but the parent is A. So the mother is A. So the mother should be having one allele which is having your IA, and the other one should be having a IO. Similarly over here we have IB and IO, isn't it? So it is because of this we have an O blood group. The individual is having O blood group, and the parents are definitely heterozygous. They are not homozygous. If the parents were homozygous, the offspring wouldn't have had an O blood group. The parent has to be an heterozygous. They should be in a heterozygous condition. Clear? So here, which is the right answer? Both mother and father are heterozygous for A and B blood group respectively. So the option is D. Option is D. Clear? If a double-stranded DNA has 20% of cytosine, what will be the percentage of adenine in it? 20%, 40%, 30%, or 60%? Now here, we know the cytosine percentage as 20. Now this is your DNA following your Chargaff's rule. What does the Chargaff's rule state? That the amount of purines is always equivalent to the amount of pyrimidines in the DNA. Why? It is always the purine which binds with the pyrimidine, or a pyrimidine with the purine. You don't get purine or pyrimidine binding. It is always a purine which binds with the pyrimidine. So the amount of purines is always equivalent to the amount of pyrimidines. And there also we know that adenine always binds with thymine and cytosine always binds with guanine, and vice versa. So if you know the percentage of one, you can find out the percentage of the other three. For example, we know that cytosine is 20%. Isn't it? Cytosine is 20%. Cytosine always binds with guanine. So if you have 20% of cytosine, then definitely the guanine percentage is also 20, because always the cytosine binds with the guanine. So 20 plus 20 is 40% of cytosine and guanine itself you have. Now what is the percentage of adenine and thymine you have? The remaining 60% is distributed between adenine and thymine. Now how did I get 60%? Out of the 100%, if you remove 40, then it is 60. So I've got 60%. Out of the 60%, this has to be divided between your adenine and thymine, isn't it? So they always bind. So the amount of adenine is always equivalent to the amount of thymine. So this is going to be divided by 2. So each one is equal to 30%. So you have 30% of adenine, 30% of thymine. So the right option that you have over here is C, 30%. Select the mismatch. Watson and Crick, DNA double helix. Frederick Misher, nuclein. Frederick Griffith, bacteriophages. Wilkinson, Franklin, X-ray diffraction studies. So Watson and Crick gave us the double helical model. So this is the right answer. Frederick Misher gave the term nuclein. This is also true. Frederick Griffith had his work on bacteriophages. No. Frederick Griffith was the one who gave us the transforming principle. He was the one who laid the basis to say that DNA is the genetic material. So Frederick Griffith did all his studies on the particular bacterium known as Streptococcus pneumoniae, which is also known as Pneumococcus pneumoniae. So he did not do his work on bacteriophages. He worked on your Streptococcus pneumoniae, also known as Pneumococcus pneumoniae. And Wilkins and Franklin did their studies on the X-ray diffraction. So which is the mismatch over here? C is the mismatch over here. Frederick Griffith, bacteriophages. In eukaryotes, heterogeneous nuclear RNA, hnRNA, is transcribed by RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, RNA polymerase 4. So here we have, in eukaryotes, we have 3 different types of RNA polymerases. RNA polymerase 1, 2, and 3. Now what does RNA polymerase 1 code for? They usually go for your large rRNA precursors, that is your 28S rRNA, 18S rRNA, 5.8S rRNAs. Whereas RNA polymerase 2, they bind for your, or they code for your hnRNA as that which is heterogeneous nuclear RNA, which is then going to be modified to the matured form as mRNA. And then we have your snRNAs, small nuclear RNAs. RNA polymerase 3 is responsible for your rRNA. Which rRNA? 5S rRNA, as well as your tRNA, transfer RNA. So the question is, which is in eukaryotes, which type of RNA polymerase is going to help in the transcription of your hnRNA? So the right option over here is B, that is RNA polymerase 2. Option is RNA polymerase 2. If the sequence of nitrogen bases of the coding strand of DNA in transcription unit is 5' ATG AATG 3', the sequence of bases in the RNA transcript would be 5' AUG AAUG 3', 5' UAC UUAC3, 5' CAUU CAU 3', 5' GUAA GUA 3'. Now here, what is asked? What is the sequence? They have given us the coding sequence, the coding strand, the sequence of the coding strand of the DNA, and you have to find the sequence on the RNA transcript. So as you already know, the DNA is double-stranded. At the time of transcription, this double-stranded DNA that is there, it will unwind, and you have two strands that you're going to get, isn't it? One will be your non-template strand. This non-template strand is not going to code into your protein. Okay, this is a non-template strand. This non-template strand is also known as a sense strand or the coding strand, whereas the other strand is a template strand, antisense strand, or a non-coding strand it is called. But actually, this non-coding strand that we call, which is your template strand, this is the one which is going to act as a template and transcribe into the RNA. Okay, so usually this template strand will be having a complementary sequence of that of your non-template strand, which is your coding strand. So definitely, when it comes to the RNA transcript that you have, this RNA transcript will be having the sequence just like that of your coding strand, because these are complementary, the coding strand and the non-coding strand. Actually, the non-coding strand is the one which is a template strand, which is going to transcribe into a RNA. So here, this coding strand that you have will be a complementary to your non-coding strand or your template strand. And this sequence that you're going to get, the mRNA transcript that you're going to get, will be exactly similar to that of your coding strand. Okay, because these are complementary. This is the one. Okay, this is the one which is going to form your mRNA transcript, the non-coding strand. This is the coding strand. So this will be similar to this. The only difference is wherever there is thymine in the coding strand, on the mRNA, you will be having your uracil, because in the mRNA, you're not having a thymine, isn't it? You have your uracil over here. So definitely, if here the coding strand is 5' ATG AATG 3', what will it be? In the your mRNA, definitely it will be 5' instead of A. So you have 5'. Instead of A, you will be having U. Sorry. You will be having U. Sorry, you will be having A over here. Wherever there is T, you will be having U. G will be G, A, U, G, 3'. So which is the right option over here? So the option is A, wherein you have 5' AUG AAU G3, because once again I repeat, your coding strand and your non-coding strand are your complementary. It is your non-coding strand or the template which is going to transcribe into the mRNA. So the mRNA will be exactly similar to the coding strand, except that wherever there is thymine in the coding strand, which is a DNA, you will be having uracil in the mRNA. So the right option over here is A, 5 prime, AUG, AAUG, 3 prime. Arrange the steps of DNA fingerprinting technique in the correct order. 1. Separation of DNA fragments by electrophoresis, digestion of DNA by restriction endonucleases, hybridization using labeled VNTR probe, isolation of DNA, detection of hybridized DNA fragments by autoradiography, transferring the separated DNA fragments to nitrocellulose membrane. So which is the correct order of sequence? So we'll just see here what is happening. We have the isolation of DNA that is taking place. First you isolate the DNA. This is followed by the digestion of isolated DNA with the restriction endonucleases, followed by the separation of restricted DNA fragments by gel electrophoresis. Then we have the blotting, that is nothing but your transfer of separated DNA fragments from the gel to a nitrocellulose or a nylon membrane, followed by hybridization, that is by using your VNTR probes, which are radio labeled or chemically tagged, and ultimately you go for autoradiography. So the sequence over here is, it begins with isolation of DNA and ends with autoradiography. So the right option that you have is A over here. Okay, A is the right option because the first step that you have is isolation of DNA. So this is first. This is first. Followed by, sorry about that. Then it is followed by the digestion. Second one is here, digestion of DNA by restriction endonucleases. This is followed by separation of DNA fragments by gel electrophoresis. Okay. The fourth is your transferring the separated DNA fragments to nitrocellulose paper or nitrocellulose membrane, and then you have the hybridization using your labeled VNTR probe, and the last step is detection of hybridized DNA fragments by autoradiography. So the right option is A over here. It is A. The most accepted line of descent in human evolution is Australopithecus, Ramapithecus, Homo erectus, Homo habilis, Homo sapiens, Homo erectus, Homo habilis, Homo sapiens, Ramapithecus, Australopithecus, Homo habilis, Homo erectus, Homo sapiens, Australopithecus, Ramapithecus, Homo habilis, Homo erectus, Homo sapiens. So here we began with the Dryopithecus, we moved on this way, isn't it? We began with the Dryopithecus, followed by the Ramapithecus, which may have been there about 15 million years ago on the Earth's surface. We believe that they were present about 15 million years ago, followed by the Australopithecus, which may have been here around 3.8 to 2.9 million years ago. Then we have the Australopithecus, Homo habilis, Homo erectus, Homo sapiens, and the modern man, Homo sapiens sapiens, isn't it? So if you see the order according to this, the right option over here is definitely your C, wherein we have the Ramapithecus, which is followed by Australopithecus, Homo habilis, Homo erectus, and Homo sapiens. So this may have been here for about 15 million years ago. Australopithecus about 3.8 to 2.9 million years ago. Homo habilis may have been here somewhere around 1.65 million years ago to somewhere 2.3 to 1.6 million years ago. Homo erectus may have been here about 1.08 million years ago. And then we have the modern man. So this is the order. So the right option over here we have is C, Ramapithecus, Australopithecus, Homo habilis, Homo erectus, and Homo sapiens. The sporozoites that cause infection when a female Anopheles mosquito bites a person are formed in the liver of the person, RBCs of mosquito, salivary glands of mosquito, intestine of mosquito. So here we have the life cycle of your malarial parasite. Isn't it? We have the life cycle of the malarial parasite. Now here in the liver of the person, the once the vector, that is your female Anopheles mosquito, bites a human being and transfers the protozoa into the male, into the human being, what happens is the sporozoites that are there, they will reach the liver. Okay, they will reach the liver, and in the liver they are going to reproduce asexually, and then they are going to be transformed into your gametophytic stages. Okay, from there, you know, merozoites and all they will form, and they'll be formed into your gametophytic stages. So here, from here, what happens is it is once again bit by another one, and it will move on. So it will go into the into your mosquito. In the mosquito, it is going to complete its life cycle. Now remember, the sporozoites that infect that cause infection when a female Anopheles mosquito bites a person are formed where? So here the option that you have over here, B, RBCs of the mosquito. This is wrong because mosquitoes do not have RBCs. They do not have RBCs. So they have hemolymph. They don't have the red blood like us. They have a hemolymph. Okay, so this is not the option, definitely. Now, where is it formed? Whether it is formed in the liver of the person, salivary gland of the mosquito, or the intestine of the mosquito. Once the mosquito bites the individual, the mosquito will be releasing the sporozoites. So definitely it is not in the liver of the person. In the liver of the person, they are going to be transformed into schizoids as well as into your merozoites. They're not going to be your sporozoites. Sporozoites are formed in the mosquito itself. So which part of the mosquito? Whether it is the salivary gland of the mosquito or the intestine of the mosquito. So it is formed in the intestine of the mosquito. Option is D, intestine of the mosquito. Smack is a drug obtained from the latex of Papaver somnifera, leaves of Cannabis sativa, flowers of Datura, fruits of Erythrosile coca. So, where do you get it? So, smack is nothing but diacetylmorphin. It is a acetylated version of morphine. Morphine is usually used as a potent painkiller. Okay, and that has been acetylated, and it has been transformed into a drug called as smack. Now, from where do you get it? So, it is obtained from the latex of Papaver somnifera. Okay, morphine is obtained, which is then transformed. From the leaves of Cannabis sativa, you get your marijuana and so on. And from fruits of Erythrosile coca, you get a drug known as cocaine. So, which is the answer over here? Definitely it is A, that is the latex of Papaver somnifera. Yeah. The substance produced by a cell in viral infection that can protect other cells from further infection is serotonin, colostrum, interferon, or histamine. So here, what is it? Is it serotonin? It is not serotonin because this is something that is secreted to carry the message from the brain, and this is responsible for your moods, your sleeping, all that is controlled by this. Colostrum, you know that it is the milk produced in the initial day of lactation, which is rich in nutrients, isn't it? It is rich in antibodies, which is given. That is why it is very necessary that the child is given, or the baby is fed with colostrum in the initial day of lactations. Interferon, this is actually the substance which is produced by a viral infected cell, and this is like a flag. It is alerting the other cell. See, I've been attacked. I have been attacked by a virus, so you better be alert. That is what it acts as a flagging. So interferon is the one, and histamine is actually produced in response to allergic reactions. Okay, so the right option over here is C, interferon. The most abundant antibody produced against allergens is IgE, IgA, IgG, IgM. Now, Ig is nothing but your immunoglobulin. So, we have IgA, which is secreted usually in your mucus, saliva, and so on. It's a secretory one. Okay, and this is mainly meant to tag the pathogens for destruction. Then we have IgD, which is your beta cell or a B cell receptor. And this actually stimulates the release of your IgM. Okay, it stimulates the release of IgM. Now, this IgM is one of the main antibody of your primary response. Okay, it is one of the main one, and it is something which remains, it is immune system memory, we call it B cell receptor and the immune system memory. We have IgG, which usually binds with your phagocytes. Okay, and this is the one, which is the main antibody that is present in response for your secondary responses in your blood. And IgE is actually meant for allergy and antiparasitic activity. So which is the most abundant one against allergens? So definitely it is IgE. IgE. Which of the following are crossed to give sugarcane varieties with high sugar, high yield, thick stem, and ability to grow in the sugarcane belt of North India? Saccharum robustum, Saccharum officinarum, Saccharum barberi with Saccharum officinarum, Saccharum sinus with Saccharum officinarum, or Saccharum barberi and Saccharum robustum. So basically, we have two types of sugarcanes, isn't it? The main two types of sugarcane, the North Indian variety and the South Indian variety. One is very thin, tall, but the sugar yield is less. The other one is a dwarf one, with a short internodes, dwarf in the sense shorter internodes, thicker ones, but with more sugar content. So definitely we want something which is tall with longer internodes, with more sugar content, which is more sweeter, where we'll get more sugar out of it. So definitely we have to cross one variety with the other, one which is tall and having more longer internode, with a shorter one having more sugar content in it. So among these, which is the one that we are going to cross? It is B. Saccharum barberi is crossed with Saccharum officinarum. Farmers have reported over 50% higher yields of rice by using which of the following biofertilizers? Bacillus thuringiensis, Azolla pinnata, Frankia, Crotalaria, gentiana. So among these, which is the one that is used in the paddy fields in order to enhance the productivity? So Bacillus thuringiensis is actually a type of bacterium which acts as an insecticide, isn't it? So which is against your specific insects like cotton ball worms and those. So this is not the one, definitely not the one. We have Azolla pinnata. Azolla is actually a nitrogen fixing organism. So we use this, Azolla is the one which is used by the farmer. So in this picture, you can see the farm which has a lot of Azolla in it, isn't? So Azolla pinnata is used as a biofertilizer. Now Frankia also fixes atmospheric nitrogen in your non-leguminous plants. Okay, whereas Crotalaria is your Fabaceae member. So among the four options, the right option that the farmers use in order to enhance the productivity of the yield, is of the rice fields, it is B, Azolla pinnata. Match column 1 with column 2 and choose the correct option. Column 1, ladybird, mycorrhiza, biocontrol agent, biogas. Column 2, Methanobacterium, Trichoderma, aphid, glomus. So here, which is the right one? Okay, so the right option over here is A, wherein the ladybird is used for the aphid population. Mycorrhizae is used. It is a mycorrhiza is an association, fungus, symbiotic association with the glomus. The biocontrol agent is Trichoderma, and then we have biogas, which is produced by a methanobacterium. So the right option over here is A. Among this, the right option is A. Gel electrophoresis is a technique of separation of charged molecules under the influence of magnetic field, incorporation of DNA molecules into the cell through transient pores made due to electrical impulses, Separation of DNA fragments through the pores of agarose gel under the influence of electric field. Separation and purification of gene products. Gel electrophoresis. What is this? This is actually the separation of DNA fragments through the pores of agarose gel under the influence of electric field. So the process of gel electrophoresis, wherein you are using the agarose gel slab in order to separate out the DNA fragments, isn't it? So here it is the separation of DNA fragments through the pores of agarose gel under the influence of electric field. The gene ROP present in PBR322 cloning vector codes for the proteins involved in translation, replication of the plasmid, synthesis of ampicillin only, synthesis of tetracycline only. So you have to find what is the function of ROP. So this is the structure of your PBR322 vector, isn't it? So here we have ROP very close to the ORI sequence. Now what is this ROP representing? Why is it present? It is mainly meant for replication. Okay, it is meant for replication. So if you're talking about ampicillin and tetracycline, we have these resistance genes over here, isn't it? We have the resistance genes. So they act as a marker actually when you try to pick out the recombinant ones. Whereas the function of the ROP is, it is mainly meant for the replication of the plasmid. So the option is B, replication of the plasmid. What is used to precipitate out the purified DNA during isolation of genetic material? Bromophenol blue, chilled ethanol, ethidium bromide, agarose gel. So here in the picture, we can see how we have spooled out the DNA. Isn't it? We are spooling out the DNA. In order to spool out the DNA from the cell, so in order to get the thread-like DNA. So here you can see, how do you get it? What are you going to use among these, which will give you the spooling out very easy? Precipitation takes place to purify. So ethidium bromide, no, definitely not ethidium bromide, isn't it? It is used somewhere else. Whereas agarose gel, as we discussed in the previous one, agarose gel is used for separation of DNA fragments under gel electrophoresis. So which is the one? The right option over here is chilled ethanol. So using chilled ethanol, we are able to precipitate out the purified DNA during isolation of genetic material. Significance of heat shock method in bacterial transformation is to facilitate binding of DNA to the cell wall, uptake of DNA through membrane transport proteins, uptake of DNA through transient pores in the bacterial cell wall, expression of antibiotic resistance gene. So heat shock. So this is wherein you are raising the temperature to 42 degrees and suddenly, so that it can be absorbed into the cell, into the host cell. The plasmid is absorbed into the host cell. So why do you give the heat shock in the case of your bacterial transformation? So the option over here is C, for the uptake of DNA through the transient pores of the bacterial cell wall. Okay. So option is C over there. Which of the following is not a genetically modified organism? GMO. Golden rice, rosy, ruppy, and dolly. Now, which among this is not? So, golden rice. Golden rice is a genetically modified plant. You know that golden rice has the precursor beta carotene. It has the beta carotene gene in it, which is a precursor of vitamin A. So, it is modified, isn't it? Using your daffodils and Irvinia and all, we have isolated the genes and incorporated into the rice embryo in order to get that golden rice. And this is meant, as a beta carotene precursor of your vitamin A, that is your beta carotene. So definitely it is not there normally. Since it is not there normally, we have genetically modified it. So definitely golden rice is also an example for your genetically modified organism. Now what is asked in the question is not a genetically, so it is not your golden rice. Next we have rosy. Rosy is a variety of cow which is a genetically modified cow. Now this cows have the presence of your human lactalbumin, alpha-lactalbumin gene in them. So they are able to produce lactalbumin in the milk. So this Rosie is also a genetically modified organism. Coming to Ruppy. What is this Ruppy? Ruppy actually means ruby puppy. Okay, so this was developed in Korea. It is also a genetically modified organism. Actually, this is a type of dog. We have the beagle breed, isn't it? Of a dog, the beagle breed. Now here what they have done with the Ruppy is they have incorporated genes, fluorescent genes in them. About 4 dogs were developed, wherein the fluorescent gene was introduced into them. So fluorescent protein actually, which was glowing red when it was subjected to UV radiation. So these are genetically modified dogs. So you have now they are trying to develop another type of dog known as doggies. Yeah, I know doggy. Doggy is also a genetically modified dog, mainly the beagle, which is very muscular. Beagles are usually not very muscular. These are small breeds of dogs, isn't it? So this was developed. Now the Parapi is also genetically modified animal, wherein they have a fluorescent protein in it, which will glow red when it is subjected to your UV radiation. Whereas coming to Dolly, Dolly was a sheep, isn't it? So Dolly is actually a clone. There is no genetic manipulation over here. It is just a clone of a cell. From the udder cell, an entire animal was developed. So Dolly is a cloned animal. She is not a genetically modified animal. So among these, the option which is not a genetically modified organism is Dolly the sheep. BT cotton is not a GM plant, insect resistant, a bacterial gene expressing system, resistant to all pesticides. So BT cotton is nothing but Bacillus thuringiensis cotton. This is a cotton which has the gene which has been isolated from the Bacillus thuringiensis. And this is toxic to your Lepidopteran and Diopteran insects. So this is definitely a GM plant because it has been modified, isn't it? It is insect resistant, yes. Cotton ball worms and all, it is resistant to it because it has that insecticidal protein within it, which has been isolated from Bacillus thuringiensis. So it is a bacterial gene expressing system, yes, because it has a gene which has been isolated from the bacterium. Resistant to all pesticides, no, it is not resistant to all pesticides. So the right option, BT cotton, is not D, resistant to all pesticides. Use of bioresources by multinational companies and other organizations without compensatory payment and proper authorization from the countries and people concerned is termed as resource partitioning, biopiracy, patenting, biofortification. So what is this? Resource partitioning. This is how the organisms will be interacting with each other, especially when there is a competition. When they are competing for the same niche, that time they will say, okay, I will be living during the daytime, you live during the nighttime. I will hunt during the day. The best example is your carnivores in a jungle, wherein their feeding habit is different because their feeding mechanism is the same, isn't it? So feeding habit is, the time of feeding is different. Some of them are nocturnal, some of them are diurnal. And similarly, on a tree, we have the birds occupying the different layers of the tree in the canopy, isn't it? And this is known as resource partitioning. If they don't go for this resource partitioning, definitely because of competition, then it is an adverse effect for them. What by biopiracy? This is the right option, wherein the multinational companies usually they try to steal without giving proper compensatory payment. Okay, so one best example for this is how US stole basmati from us and they marketed it under their own name, isn't it? Then we filed a suit against them and we won anyway because we know that basmati is us. So biopiracy is the right answer. So patenting, what do you mean by patenting? Someone has invented something or done something they can get the patenting right, saying that this is mine. They have to go and register themselves and say this is mine. Nobody can use this unless they have a permission from me. Okay, this is what patenting is. And biofortification is a process of enhancing the nutrient content of the crop plants. So which is the one where the multinational companies and organization try to steal from the countries? It is your biopiracy. Answer is B, biopiracy. Which of the following is an important adaptation of animals to cold climate? Thin layer of body fat, estivation, increased tendency to shiver, reduced surface area to volume ratio. So, what is it? Which of the following is an important adaptation to cold climate? Now, thin layer of body fat, no, they should have a thick layer of body fat, because body fat that is there, it acts as an insulator. So when a very cold climate is there, if they have a very thin layer, the survival chances are less. That is why organisms, the animals living in colder climate, usually have very thick fat layer. Estivation. It is not estivation, because estivation is summer sleep. Okay, hibernation is winter sleep. This is summer sleep. This is what they become inactive. They just go to sleep during summer. That is in order to avoid the very high temperature. Increased tendency to shiver. Yes, when it is cold, we start shivering. That is a mechanism. It's a homeothermic mechanism. But this is not one of the very important adaptations. You will not see the polar bears and the other penguin all the time shivering like this. They have their own adaptations, isn't it? Do you find them all the time shivering and going around? They don't. That is because there is an the shivering is there, but it is not that. They have been adapted to it internally. Reduce the surface area to the volume or volume ratio. So this is what the Allen's rule states, isn't it? In according to Allen's rule, you all Allen's rule and already cultivated. So Allen's rule ಅಲ್ಲಿ ಏನಿದೆ, ಈ colder climate in animals ಅಲ್ಲಿ usually ಸಣ್ಣ ಕಿವಿ ಮತ್ತೆ ಸಣ್ಣ ಕಾಲು ಇರುವುದಿಲ್ಲ, isn't it? So usually ಇರ್ತದೆ. I'm really sorry. ಇರ್ತದೆ. So they have shorter ears and shorter limbs in colder climate. So here you can see the rabbits, the different types of rabbits. The rabbit which is there in the hot climate usually has longer ears, isn't it? Whereas the rabbits in the colder climate have got very short ears. So similarly, we have the Arctic fox and the gray fox also. If you see a lot of differences. So their surface area, extremities are usually reduced in order to conserve heat. So Allen's rule is applicable over here. So this is the right answer. They have reduced surface area to volume ratio. A population has more older individuals than younger ones. What would be the status of the population after some years? Decline, stable, expand, or steady? So here we have the three types of population, isn't it? We have the expanding, stable, and the declining population. The yellow one represents the young individuals, that is the pre-reproductive age individuals. The bluish green that you can see here, this is your reproductive age one, and the pink one represents the senescent phase. So whenever the pre-reproductive stage, that is your young ones, the number is more than the other, this shows that the population is an expanding population. When the number of your pre-reproductive and the reproductive, that is the young ones and the reproducing organisms, the number is almost the same, then it is known as a stable or the steady population. And when the number of individuals in the pre-reproductive stage or your young ones is less, then when compared to the others, then it is known as a declining phase. So here what is given has more individuals than the younger ones. So what type of population do you see after the status of the population after some years? So definitely, since the number of older individuals is more than the younger ones, the population will be declining population. So the option over here is A. Clownfish living among the stinging tentacles of sea anemone is an example for competition, predation, mutualism, or commensalism. So we know that competition is where both the individuals that are interacting will be having a negative effect on each other. In predation, one will be benefit and the other one will always be in the disadvantage. When it comes to mutualism, we have both are benefited, both organisms. This is a type of interaction between the species, isn't it? Between the organisms. So both are going to benefit. When it comes to commensalism, one is benefited and on the other, there's neither any benefit nor harm. So when you come to your sea anemone, okay, the clownfish, along with your we have the clownfish and the sea anemone interaction. If you see, this is actually an example for commensalism, because this sea anemone, whether it is not the sea anemone, the clownfish, whether it is there or not, residing between the sea anemone, it doesn't make a difference to this metridium or your sea anemone. It doesn't make a difference. So one, it is, yes, it is going to get a place to hide, whereas the other one, whether it is there or not, it is of no use. So the right option, the type of interaction that you see over here is D, commensalism. The correct sequence of stages of succession on a bare rock is lichens, mosses, grasses, shrubs, trees. Lichens, grasses, mosses, shrubs, trees. Lichens, mosses, shrubs, grasses, trees. Lichens, grasses, shrubs, mosses, trees. So what is the right interaction? So we know that always on a bare rock, the pioneer species is a lichen. Isn't it? It begins with the lichens, and from the lichens, how it is going on. So you are, and ultimately, as a stable community, you will be having the trees. So which is the one? Which is the order of succession over here? So it is the lichens, followed by the mosses, then the grasses, the shrubs, and the trees. So the right option over here is A. Lichens, mosses, grasses, shrubs, and trees. If 10 joules of energy is available at the producer level, then amount of energy present at the level of the secondary consumer is 100 J, 10 J, 1 J, 0.1 J. So this problem is actually based on the 10% law. Okay, so as you're moving from one tropic level to the other, only 10% of energy is passed on to the next tropic level. Remaining 90% is lost. So here we are saying that 10 joules of energy was available at the producer level, isn't it? It was here at producer level. We had 10 joules over here. So as it is moving up, only 10% is carried on to the next one. Since it is only the 10% that is carried on, when it is going on to your primary consumer, it is only 1 joule. From primary consumer, when it is going on to the secondary consumer, it is 0.1. And from there to the tertiary, it is going to be 0.01 joules, isn't it? So among these, definitely it is not 100. It cannot be 10 because it is following your 10% law. It cannot be 1 joule because 1 joule is what you get the amount of energy at primary consumer. We have asked about your secondary consumer. So the right option over here is D, 0.1 joule. So this is basically based on the 10% law, stating that only 10% of the energy is transferred to the next tropic level, remaining 90% is lost. Who confirmed communities with more species tend to be more stable than those with less species? Alexander von Humboldt, we have David Tillman, Paul Ehrlich, and then we have Edward Wilson. So who among them? So all of them have contributed towards biodiversity, isn't it? Among these, who confirmed that the communities are, that with more species tend to be more stable as than those with the less species? So among these, it was David Tillman, B, who proposed this, that communities with more species tend to be more stable than those with less species. What is common to the seed banks, tissue culture, and cryopreservation? All are in situ conservation method, all are ex situ conservation method, all require ultra-modern equipment and very large space, all are methods of preserving extinct organisms. So here this is based upon in situ and ex situ means of conservation. What do you mean by in situ means of conservation? In situ means of conservation andre ಅದಿರುವಂತಹ ನ್ಯಾಚುರಲ್ ಜಾಗದಲ್ಲಿ, ಅದು ಆಕ್ಚುಲಿ ಇರುವ ಜಾಗದಲ್ಲೇ ಅದನ್ನು ನಾವು ಕನ್ಸರ್ವ್ ಮಾಡೋದು, ಅದನ್ನು ಪ್ರೊಟೆಕ್ಟ್ ಮಾಡುವಂಥದ್ದು. ವೆರಸ್ ವೆನ್ ಯು ಟಾಕಿಂಗ್ ಅಬೌಟ್ ಎಕ್ಸಿಟಿವ್ ಕನ್ಸರ್ವೇಷನ್ ಅಂದ್ರೆ ಅಲ್ಲಿಂದ ಅದರ ಒರಿಜಿನಲ್ ಜಾಗದಿಂದ ತೆಗೆದು ನಾವು ಬೇರೆ ಕಡೆ ಇಟ್ಟು ಅದನ್ನ ನಾವು ಕನ್ಸರ್ವ್ ಮಾಡುವುದಂತ, ಅದನ್ನು ಪ್ರೊಟೆಕ್ಟ್ ಮಾಡುವಂಥದ್ದು. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಈಗ ಝೂಸ್, ಪಾರ್ಕ್ಸ್, ಗಾರ್ಡನ್ಸ್ ಇದೆಲ್ಲ ನಮ್ಮ ಎಕ್ಸಿಟಿವ್ ಕನ್ಸರ್ವೇಷನ್ ಗೆ ಬರ್ತದೆ. ನಿಮ್ಮ ಆರ್ಬೋರೇಟಮ್ಸ್ ಇದೆಲ್ಲ ಎಕ್ಸಿಟಿವ್ ಕನ್ಸರ್ವೇಷನ್ ಅಂದ್ರೆ ಅದು ನ್ಯಾಚುರಲ್ ಪ್ಲೇಸ್ ಇಂದ ನಾವು ಬೇರೆ ಕಡೆ ಇಟ್ಕೊಂಡು ಹೋಗುವಂತ. ಇನ್ಸಿಟಿವ್ ಕನ್ಸರ್ವೇಷನ್ ಅಂದ್ರೆ ನಮ್ಮ ಬಯೋಸ್ಪಿಯರ್ ರಿಸರ್ವ್ಸ್, ಅಲ್ಲಿ ಅಂದ್ರೆ ಅದರಲ್ಲಿ ಇರುವಂತಹ ನ್ಯಾಚುರಲ್ ಹ್ಯಾಬಿಟಾಟ್ ಅಲ್ಲೇ ಆ ಗಿಡ ಮರಗಳನ್ನು ನಾವು ಸಂರಕ್ಷಿಸ್ತಾ ಇದ್ದೇವೆ. ಸೊ ವಿ ಹ್ಯಾವ್ ಇನ್ಸಿಟಿವ್ ಅಂಡ್ ಎಕ್ಸಿಟಿವ್ ಮೀನ್ಸ್ ಆಫ್ ಕನ್ಸರ್ವೇಷನ್. ಸೊ ಇದರಲ್ಲಿ ಸೀಡ್ ಬ್ಯಾಂಕ್ಸ್, ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಅಂಡ್ ಕ್ರಯೋ ಪ್ರಿಸರ್ವೇಷನ್ ಇದು ಯಾವುದು? ಅದು ಅದರ ನ್ಯಾಚುರಲ್ ಜಾಗದಲ್ಲೇ ನಾವು ಅದನ್ನು ಪ್ರೊಟೆಕ್ಟ್ ಮಾಡ್ತಾ ಇದ್ದೇವೆ, ಇಲ್ಲ ಬೇರೆ ಕಡೆ ಹೋಗಿ ಅದನ್ನು ಪ್ರೊಟೆಕ್ಟ್ ಮಾಡ್ತಾ ಇದ್ದೇವೆ. ಸೊ ಡೆಫಿನೆಟ್ಲಿ ಓವರ್ ಹಿಯರ್ ದೀಸ್ ಆರ್ ಯೋರ್ ಎಕ್ಸಿಟಿವ್ ಮೀನ್ಸ್ ಆಫ್ ಕನ್ಸರ್ವೇಷನ್. ಆಪ್ಷನ್ ಇಸ್ ಬಿ, all are exitive conservation methods. Moving on to the last question. Minamata disease was caused due to the consumption of seafood contaminated with cadmium, mercury, pesticides or selenium. So here your mercury, cadmium, these are all heavy metals. Okay, they tend to get in ನಿಮ್ಮ ಬಾಡಿ ಒಳಗೆ ಅದು ಎಂಟರ್ ಆದ್ಮೇಲೆ ಅದು ನಿಮ್ಮ ಮಸಲ್ಸ್ ಅಲ್ಲೆಲ್ಲ ಡೆಪಾಸಿಟ್ ಆಗ್ತದೆ, ಅದು ಬಾಡಿಯಿಂದ ಎಲಿಮಿನೇಟ್ ಆಗುದಿಲ್ಲ. ಅಂಡ್ ದೀಸ್ ವಿಲ್ ಬಿ ಲೀಡಿಂಗ್ ಟು ಡಿಫ್ರೆಂಟ್ ಡಿಸೀಸಸ್. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಕ್ಯಾಡ್ಮಿಯಂ ಪಾಯ್ಸನಿಂಗ್ ಇಂದ, ಇದು ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ ಕ್ಯಾಡ್ಮಿಯಂ ಪಾಯ್ಸನ್ ಡೆಪಾಸಿಟ್ ಆಗೋದ್ರಿಂದ ಕಿಡ್ನಿ ರಿಲೇಟೆಡ್ ಪ್ರಾಬ್ಲಮ್ ಸ್ಟಾರ್ಟ್ ಆಗ್ತದೆ. ಸೊ ದಿಸ್ ಇಸ್ ದ ವೆರಿ ಕಾಮನ್ ಡಿಸೀಸ್ ಇಸ್ ಇಟೈ. ವೆರಾಸ್ mercury poisoning from mercury poisoning we suffer from something known as Minamata. So Minamata was actually a small village located on the banks of river Minamata in Japan, wherein the people started consuming, they ಅಲ್ಲಿ ಮೇನ್ ಅದ್ರದ್ದು ಅವರ ಫುಡ್ ಅಂದ್ರೆ ಮೀನ್ಸ್ ಆಫ್ ಇನ್ಕಮ್ ಅಂದ್ರೆ ಅಲ್ಲಿರುವಂತಹ ಮೀನುಗಳು. ಆ ಮೀನು ಆಕ್ಚುಲಿ ಹತ್ತಿರದಲ್ಲಿರುವ ಫ್ಯಾಕ್ಟರಿಯಿಂದ ಬಂದಿರುವಂತಹ ರಿಲೀಸ್ ಆಗಿರುವಂತಹ ವೇಸ್ಟ್ ಇಂದ ಏನಾಗಿದೆ, ಆ ಮೀನುಗಳಿಗೆ ಮರ್ಕ್ಯುರಿ ಪಾಯ್ಸನಿಂಗ್ ಆಗಿದೆ. ಆ ಇಂಡಿವಿಜುವಲ್ಸ್ ಅದನ್ನು ಕನ್ಸ್ಯೂಮ್ ಮಾಡಿದಾಗ ಅವರ ಬಾಡಿಯಲ್ಲಿ ಅದು ಎಂಟರ್ ಆಗಿ they started showing certain particular sign and symptom, and the name of the disease was named as Minamata disease. Selenium poisoning ಇಂದ ನಮಗೆ ಸೆಲೆನಿಯೋಸಿಸ್ ಆಗ್ತದೆ. ಸೊ ಈ 4 ಆಪ್ಷನ್ಸ್ ಅಲ್ಲಿ out of the 4 options the Minamata disease was caused by mercury poisoning. So the right option over here is mercury poisoning. Okay, so here we come to the end of 60 questions. Hope this will help you in your competitive examinations. Thank you.